introduction. And for the next several lectures, what we'll be doing is then looking at the ectoderm, mesoderm, and endoderm, and asking the question of what do these individual germ layers eventually become in the embryo. So we're at the point now where we've gone through all the early stages, we've finished gastrulation, we've probably established at least some body patterning. So what's the next big thing for the embryo to do? And we're going to start out by talking about ectoderm-derived structures. So in a way, if you just sort of kind of generally remember the order of things that we've talked about in this course, you'll say, oh, that was ectoderm, or this middle portion is all mesoderm, and it's all endoderm at the end. So maybe that will help establish that long-term connection. Okay, so first thing to talk about is the development of the central nervous system, which of course is going to become the brain and the spinal cord. How this starts. So here's our vertebrate embryo following gastrulation, and at this point now there's been a substantial rearrangement of tissues with the mesoderm sandwich in between the internal endoderm and the outer ectoderm. And one of the first things that's happening now is we're going to go through neurulation and form the neural tube. So we did talk about this a little bit before, but just to refresh your memory, up here at the animal pole of the embryo, you start to see a flattening out and then a bulging up of cells. So we're starting to form here the two neural ridges. We have a little bit of mesoderm tissue that's going right down the center of the embryo, anterior to posterior, and that's going to form a mesoderm structure known as the notochord. So it's just a convenient landmark for us at this point. And as time goes by, we see then the outer ectoderm tissue start to fold in, making the neural folds, and then it eventually will pitch off and make a tube, a neural tube running the entire anterior posterior length. Okay? And then the neural crest cells are more individualized cells in between this stuff. So let's look at this neural tube. And initially, it's only about 1 cell layer thick. So it's not a very elaborate structure. And depending on the type of organism, it may sort of zipper shut from one end to the next, or it may pinch shut and seal off at several different points. As soon as this happens in the very anterior portion of the embryo, the next thing that we'll start to see is some expansion, especially of the interior volume of this neural tube. And there becomes these 3 partitions, the forebrain, midbrain, and hindbrain. And as time goes by, here's showing you a few different stages for human development at week 4, week 5, how these different regions within the anterior neural tube start to expand and change their shape. So there's still really only 1 cell layer thick, but especially this interior lumen is expanding a lot, and the constrictions are appearing between these 3 different territories. So these are going to be really critical for the future division of territories and cells and patterning and shaping of the whole brain structure. Already starts here in the very early embryo. It's also kind of interesting that different species within vertebrates are really, really similar in the way that they approach this whole next task of building brains. So I don't care if you're a zebrafish, a chicken, a rat, or a human. At this stage, the brain development looks almost identical. Just these 3 basic vesicles: forebrain, midbrain, and hindbrain. So just some terminology here. The forebrain is more officially called the prosencephalon. The midbrain will be the mesencephalon, and the hindbrain the rhombencephalon. And these 3 bulges are known as vesicles. So to start out, we're going to have 3 primary vesicles. And then if we look just a little bit later, the 3 vesicles have expanded to 5 vesicles. And they also have names that I would like for you to know. And then you can see how the adult derivatives map onto these 5 different areas. So that's really one reason for knowing about these, being aware of them, naming them, is that each of these specific structures in an adult can be tracked back to cells that have originated already at this 5-vesicle neural tube stage. So I'm not going to ask you to memorize what adult derivatives match to what particular chamber here, but I would like for you to know the chamber names. So let's focus in on the hindbrain in particular, or the rhombencephalon. And as time goes by, we can start to see some additional partitions appear within the hindbrain or rhombencephalon. And these are called rhombomeres. And there are different segments or compartments within the hindbrain. And so here's an embryo over here where you can see the head and the eye, the midbrain, and within the hindbrain, these 7 different regions or territories known as rhombomeres. And again, they each have these very particular types of structures or nerves that are going to be patterned so that they always come out of the same rhombomere from one individual to the next. So already some specification and determination is starting to happen in these cells at this early stage. And it may kind of remind you of segments within the Drosophila fruit fly and how that organism met the challenge of body plan by dividing up different physiological compartments and then expressing different Hox genes in those compartments. And, you know, mammals are not insects, they don't really do it in exactly the same way, and yet there's kind of a certain parallel here of dividing this thing up into different compartments, and then one compartment has a particular job to do for cells as they develop. And if you look at what Hox genes are expressed in these 7 different rhombomeres, you can really see some distinct patterns here. So here are different Hox genes that will be expressed in particular rhombomeres. And then they have a fairly sharp boundary as to where that expression will end. So each different rhombomere is getting a different combination of Hox genes. And this totally fits with our idea of the Hox code hypothesis and how you're getting these different transcription factors being expressed in different regions, and then that sets the cell fate for those cells in that particular territory. So here are some additional signaling molecules. It's not just Hox genes, but there are other key players as well. Here's a retinoic acid receptor, and then even a growth factor here. So growth factors and Hox genes continue to be important for brain development. Here's an in situ hybridization at an early stage or a later stage for a gene called CROX20. And this happens to be a gene that is not in one of the 4 homeobox gene clusters, but just happens to have a homeobox in it. So it is considered a member of the homeobox family, but it's in on a different chromosome, not part of one of those original sets that duplicated. And it has this interesting expression pattern early on that you'll see it in rhombomeres 3 and 5. And initially that's kind of a fuzzy looking expression. You know, you can tell it's in those territories, but it's not really crisp. Whereas at 8 hours later, it has this remarkable banded pattern within the embryo, and that really reflects the way that compartments have been set up here that are distinct and express different types of genes. And so CROX20 is one that happens to be expressed in 2 of these. Perhaps why it gets more distinct is you're now getting more elaborate regulation of the CROX20 gene, just like we saw for some of the parallel genes that are expressed in stripes. But I think probably also there's some anatomical basis for this because the cells just become physically more compacted and discrete within these territories. So in the case of humans, we have a relatively large brain compared to our body mass, and that brain takes a while to grow. So you'll still see a fair amount of extensive growth and cell division within the human brain, even after birth, and occurring up to a couple of years. So here, this dot is representing the point where birth happened for a chimpanzee and a human. And prior to that stage, when they were in the body, they're fairly similar in terms of the ratio of brain weight to body weight. But then afterwards, the chimpanzee, its brain is still going to grow somewhat between newborn and adult, but the human, it's a much more dramatic increase in brain size even after birth, which reflects our better cognitive abilities. Okay, so now I want to think about the neural tube, which is running the anterior-posterior length of the embryo, and how that tube acquires patterning on the dorsal ventral axis. So dorsal is going to be up on the top of the embryo along its back side. Ventral would be more in the belly. And one of the genes that was originally found in Drosophila, a segment polarity gene, is named hedgehog. And this gene in Drosophila, if you mutate it, it actually has a bristle phenotype in some of the weaker mutations. So you get these adult flies out that have bristles that are really short and stubby. And so they named it hedgehog because it was kind of like, hedgehogs have these bristly appearance and these thick, short bristles. Anyway, this turns out to be a mutation in a segment polarity gene. So it was one of the early patterning genes, and it was expressed in these whole array of stripes here, either in anterior or posterior halves of parasegments. And it turns out that vertebrates also have a hedgehog gene, and one of these, they have actually 3 of them, and one of them is named Sonic Hedgehog. And the way that this got its name is after a video game. Have any of you played this? See, you guys know what it is. I have no clue, but I know it's a video game. And so somebody was being cute, so I don't know, they decided because it was a parallel gene to hedgehog that we knew about in Drosophila, and then we found it in the human, they said, let's call it Sonic Hedgehog. And, Sonic Hedgehog is also a band, turns out, in Finland. So I don't know why they named themselves Sonic Hedgehog. It probably is after the video game and not after the gene, I'm guessing. And here's their album cover with the little hedgehogs on their sign, and this is in Finland, so there's some snow, and I hope they realize a car is coming and they're going to get that picture done quickly. So, anyway, so Sonic Hedgehog is a paracrine factor, and it's needed in the embryo at this early stage to help pattern the neural tube. And so the scenario that we have here is the neural tube, and the portion of that tube that's closest to the outside of the embryo, we term that the roof plate. So it's basically just the dorsalmost portion of the neural tube as it's going to develop. That's going to be in contact with the outside layer of ectoderm that's surrounding the entire embryo, and you're going to see signals coming from the ectoderm onto that roof plate and beginning to establish a territory in that roof plate that's sort of unique and special and will eventually become the dorsal side of the neural tube. So here are some of the signals. They're all TGF-beta family members, such as BMP4 or BMP7, and they help establish a unique patterning to the roof plate. Down on the opposite side of things, we have the floor plate, which is on the ventral side of the neural tube, and that's going to be closer to the notochord, which is running ventrally just below the neural tube, all along the anterior and posterior length of the embryo. And so the notochord is what's making Sonic Hedgehog here, which is a paracrine factor. So the notochord makes it, it just diffuses over and encounters the floor plate cells of the neural tube, and begins to set that up as a territory that would become ventral neural tube. So initially, as things first start out, we have these 2 external sources of induction signaling molecules. But then eventually, the neural tube cells themselves become secondary sites where they also make further paracrine factor. So up here in the roof plate, now we get these cells, after being exposed to BMP, they start to make their own BMP and reinforce and further elaborate that signal. And same thing in the floor plate. These were first exposed to sonic hedgehog, and now they're making their own and become a second source of sonic hedgehog for that neuron. So what we're going to see here is two gradients of paracrine factors and signaling set up on either side of the embryo and a neural tube. And as this plays out, it looks something like this. And the color coding is nice because it does give you the idea of the different concentrations for these two different factors. And you can see here with a rainbow of colors between these two factors, all of these different unique cell fates I'm reading out. So the very high sonic hedgehog fates will become floor plates, and then as we taper off to a little bit less sonic hedgehog, but slightly more BMPs, we get a variety of different neurons type cell fates, and then so a unique type of fate corresponding to each particular unique territory as set up by these two opposing gradients. So this mechanism, it worked really well for Drosophila when we had bicoid and nanos, and here we are with a vertebrate. Different time, different place, different species, but they're using the same kind of tools to accomplish the goal of dividing up the territory so that different fates can arise at specific spots. Okay? So that's the principle of this slide. You don't need to know the names of all these different things. So then, with each of these smooth gradients, we're going to see a response in terms of cell fates that is more of a chunked out type of distribution of cells. So some of the cells that if you look at in the neural tube at a slightly later time point, they're already being specified to these different cell fates, and they're expressing markers that are appropriate for that. So up there at the roof plate, those cells are expressing a transcription factor known as PAX7, and that is helping to pattern their fate as those particular kinds of neurons. So, really fairly discrete grouping of cells just based on whether or not these first two paracrine factors were present and in what different amounts. So here is a notochord expressing sonic hedgehog, and you can see above it the different responses of neurons within the neural tube. So from here up to the top is all neural tube. Okay, so essentially what we have here with this kind of data, looking at where different genes are expressed, is basically show it type of information. And we can also do some experiments to try to test whether or not we're accurate in the hypothesis that sonic hedgehog is the key molecule coming from the notochord that is impacting these changes within the neural tube. So it's really kind of logical experiment to do would maybe be to take an embryo and put an extra piece of notochord somewhere else to expose other portions of the neural tube to notochord or sonic hedgehog signaling. So here's that scenario, and that little brown blob over there, that's notochord tissue that's now placed laterally beside the neural tube and allowed to do its thing, allowed to emit sonic hedgehog. It'll be exposing the nearest cells to sonic hedgehog. And normally those cells should just be forming these interneuron cell fates. And in this case though, they were actually able to change and respond to the high concentration of sonic hedgehog and now take on these green cell fates, which are indicating floor plates for this embryo. And then not only that, but looking laterally, as you start to taper away from this ectopic source of sonic hedgehog, now you get some sort of medium concentration or even low concentration response fates coming out of that, such as these blue cells here, which are responding to, say, a medium level of sonic hedgehog. Was there a question? So, yeah.
So A would be wild type then?
Yeah, so A is a wild type situation, and B is a manipulated situation where we have this donor notochord It's going to establish new floor plate and new secondary motor neurons. Okay, so once you're able to do some experiments like this would be a move-it experiment, then you can start to ask questions about what are the important factors, are those factors sufficient, are those factors necessary? So, we could also look at a sonic hedgehog mutant and maybe predict that we're going to lose some of these ventral cell fates, assuming the embryo can survive to that stage. So, if sonic hedgehog is a morphogen for the ventral neural tube, kinds of things that it would be doing is going to be to manipulate these different targets within the cell. So sonic hedgehog is a paracrine factor, it's external to the cell. It's going to need to bind to a receptor. That information get transduced across the cell and then enter into the nucleus. And ultimately, we see differential gene transcription. So here's another parallel to what was going on with Drosophila. When we had these different gradients and we said that the gap genes were responsive to different thresholds of a particular morphogen. So some gap genes need at least this much or higher to turn on, and you can find genes that need, at least this much sonic hedgehog or more to turn on. Or you can flip the thing around and have it be repressive. So if you have too much sonic hedgehog, that can limit also where some of the genes will be expressed. So you see both positive and negative interactions listed here, negative and positive. So then this all leads into different expression on the part of these cells. So in the case of roof plate, the key factors here are several, and these include BMPs, dorsalen, and activin, and really all of them are members of the TGF-beta superfamily. So you could do similar sorts of experiments here, where now you're going to take a chunk of ectoderm tissue and place that in a new location and see how the fates of the neural tube respond to that particular source of signals. So here's a snapshot through this neural tube. And believe it or not, this actually is a 1 cell thick layer of cells. And those little fat portions of the cell there, this is where the nucleus and cytoplasm is. And then each of these cells extends a process that goes all the way down to one side of that neural tube region, and then another process up to the top. And amazingly enough, they actually shift where this nucleus is located over time. So it'll migrate up and down that stalk, and it does this as part of its process of getting ready to go through cell division. So at any given time, if you just look at a snapshot through the neural tube layer of cells, you'll see these nuclei at all different places. So diagrammatically, that looks like this, where here's one cell extending the entire width of the neural tube, and that nucleus and cytoplasm moving around. So when the cell is ready to divide, it's going to send the nucleus down towards the lumen portion of the neural tube. So what we're looking at here is just one cross-section through half of the neural tube. Then that cell will divide. It goes through a regular mitosis. And neuroscientists like to call this the birthday of the neuron. So at that point, we now have two cells that came from this original daughter cell. One of them will stay in the neural tube and act like a stem cell. So it'll stay in the neural tube and go through some additional cell divisions and continue to pump out more daughter cells. One of the daughter cells is going to migrate out to further more peripheral locations and start to differentiate as a particular class of neuron or glial cell. So they're going to then be located external to this neural tube. So as time goes by, you get more and more waves of cells that have been born in the neural tube and are migrating outward to more peripheral layers. So let's see what happens to them. So In these diagrams, what you're looking at is just one section through the neural tube is diagrammed by that box up there. And so here is the lumen of the neural tube on this side. And here are cells that are in that ventricular zone or in the actual neural tube portion itself. And they're going through these rounds of cell migration. As the daughter cells start to leave, they're going to migrate out to an external layer. And they're quite organized about how they arrange themselves. So they'll form one region where all the neuron cell bodies are located together, and another region where their axons and dendrites are just extending outward. So there's no nuclei in that portion. And this organization gives a kind of two different colors to the structure. And the more gray area is called the mantle, or the intermediate zone. And then out there with all the axons, that's called the marginal zone, and it's more white in appearance. So this already is evident in humans by 5 weeks. So we've gone through gastrulation, gone through neurulation, and now these cells in the neural tube are starting to proliferate and divide. And then along the length of that neural tube, what happens in the part of this process establishes whether it's going to become spinal cord or forebrain or cerebellum or whatever. So in the case of the spinal cord, here you see ventricular zone, intermediate zone with all the cell bodies, marginal zone with all the axes. And as time goes by, looking at the whole neural tube as a group, here we see what would have been the original neural tube in the center, and then all of these daughter cells that have migrated out. The dark area is their nuclei, the pale gray area is the marginal zone, and this is, a classic section through a spinal cord of the adult mammal. We have more of a butterfly-shaped structure. And it all tracks back to how this was set up during development. And in fact, you could revisit what neurulation looks like with this sort of endpoint pattern in mind. And so what they've done here is just color code these different regions. What was the region that was right in the center that would form the neural groove? And then what were the regions kind of on the ridge that met the upper portion of the neural tube in orange? And so that gave a neural tube that we might color code like so, based on the origin of these cells. And then if you look and see what those daughter cells ultimately become, it's quite interesting, because then in the adult they get to be localized in different discrete regions. The green ones are down in this region called the basal region, and that's where you'll see your motor neurons develop. And they have the job of transmitting information to the body. Whereas different group of neurons located kind of in the same territory but more dorsally, those are going to be the alar region, and they'll be receiving sensory input. So what becomes a really critical patterning for the adult organism, again, that tracks back to some really early stage of development where it first got laid out. Yeah?
Is that supposed to be basal with an A instead of an E?
Yes, probably.
Okay. Just making sure.
Like that. Okay. So, and then just a sort of a background slide here, the central nervous system. Of course, when we talked about that, we mean the brain and the spinal cord, shown in red in the diagram. And then we also have the peripheral nervous system, which would include structures outside the CNS, so nerves and ganglia, and then two different types of neurons, sensory neurons bringing input into the CNS, and motor neurons taking impulses from the CNS to move muscles or glands or other tissues. Okay, what about the cerebellum? So the cerebellum is a region of the brain that is found sort of the lower back. And this region of the brain has a lot to do with sensing movement and timing. And I always think of birds when I think of the cerebellum, because birds have to fly around in three-dimensional space, and they have to go into trees, which have a lot of twigs in them and branches, and the birds never seem to collide. They somehow manage to fly through all these structures and land on the branch, and they don't have any trouble. So I think they must be very well developed when it comes to the cerebellum. And this is all going to start the same way as it did for the spinal cord, but the types of cells that differentiate from the various different layers would be different and unique to the cerebellum. So you're going to see successive waves of cells migrate out. First they may establish an intermediate zone, the next layer out establishes the granule cell layer, and then more and more cells keep migrating out past those earlier ones and setting up yet additional layers. So in general, you'll always see the newer born cells, the latest, the most recent cells, they're going to migrate the farthest. Because it's as if everybody is born in the inner city and they have to move out to the suburbs to build a house. And so the first guys that are born, they just get to go a short ways and build their house. And then later, more and more kids have to go farther and farther out, away from the city center, to find the room to build a house and establish a location. So in general, it's sort of a, very much a directed and organized sequence of events to how these different layers. But the cerebellum has one interesting exception to this theme that I want to point out. So one of these groups of cells, known as the granule cell layers, they initially are set up as I've described, where the neuroblasts or the precursor daughter cells from those cell divisions leave the neural tube. They go out, they make a layer, it's called the granule cell layer. It's actually called the external granule cell layer. Then they're going to go through another round of mitosis, and then these daughter cells migrate backwards towards the neural tube and set up another layer of their own called the internal granule layer. So they actually go out farther, divide, turn around, and come back, which is against the theme of things, but can happen, and happens here for these cells in the cerebellum. So this type of cell movements and coordinated cell birth, migration, etc., this does a tremendous job for the brain as it starts to develop.